Good morning, everybody, or evening or afternoon, wherever you are. Welcome. Thank you again for joining us today for the Next Generation Scientist session. I'm Melissa Peppa. I'm at the University of Memphis, and I'll be co-moderating with Marcus Goncalves, who's an endocrinologist and a basic scientist at Weill Cornell Medical College in New York. It's our pleasure to be here and moderate this session. A few housekeeping notes before we get started. All of our talks are going to be recorded and will be available within approximately 5 business days after the conclusion of the conference. During the sessions, please make sure that you post all of your questions in the Q&A box so that we can make sure that your questions are answered either during the Q&A session or after the Q&A session. We have enabled the chat function for you to be able to engage with attendees, share your comments during the meeting. And if you need any technical help, you can use the chat function in Zoom to direct your questions or problems to our administrators. And one of the staff will help you and respond to your message.
So let's get started. The Next Generation Researchers session is designed to promote upcoming and young faculty researchers, and the speakers were chosen from the submitted applications. The objectives are to recognize early phase career clinicians and scientists who have committed their clinical practice and research labs to cancer cachexia, to provide an opportunity for junior investigators to discuss their clinical trial, translational, and basic science research. And the structure of the session will be the speakers will talk for 10 minutes, followed by a 5-minute Q&A, and 10 minutes goes by quickly, so we'll try to keep everybody on track and collate the questions to the end. Our first researcher is Lindsay Anderson from the University of Washington, discussing whole body and adipose tissue metabolic phenotype in cancer patients. So take it away, Lindsay.
Thank you. So I want to start with a brief overview of the interplay between some of the important pathways for affecting adipose wasting in the setting of cancer, even though I want to remind you that these mechanisms are primarily derived from preclinical data. But inflammation, in particular from cytokines like TNF and IL-6, are thought to promote lipolysis in adipocytes through upregulation or activation of lipases such as ATGL and HSL, which cleave free fatty acids, and those can be measured in the circulation as a marker of lipolysis. But in addition, IL-6 and tumor factors or things at various aspects of that are secreted by the tumor, such as parathyroid hormone-related peptide, can also promote white adipose browning by upregulation of UCP1, which is the signature marker of brown adipose that's responsible for that tissue releasing energy as heat. Although if this is occurring in humans to a meaningful extent, then theoretically we should be able to measure that at the whole body level by increased energy expenditure. But there is very little data in humans at this point to actually verify that white adipose browning is really occurring. And I want to highlight 2 observations here from the same lab, actually, that included some very brief human data at the end of some very extensive preclinical reports that they published. And what they observed here were that UCP1 was detectable in 7 out of these 8 cancer patients who were losing weight. And you see there are a whole number of different tumor types and different locations where that adipose was obtained. But this was in comparison to subcutaneous white adipose collected from 10 weight stable colon cancer patients where UCP1 was not detected in any of those patients. So they're suggesting that there's a potential relationship between cancer-induced weight loss and browning via UCP1 expression. And similarly, they, in another preclinical paper where they reported this kind of human data at the end, they are showing that patients with PTHRP that is detectable in the circulation was those patients had significantly higher energy expenditure relative to lean mass compared to those cancer patients with undetectable PTHRP in the circulation. So again, they're sort of making this suggestion that there is a relationship between PTHRP anyways in the circulation and maybe hypermetabolism. Another report linked cancer-related weight loss to inflammation in the circulation and lipolysis and browning in the adipose tissue. Here they report that circulating free fatty acids were directly associated with circulating cytokines in what they define as early and late cachexia, which is essentially the severity of weight loss in the prior 6 months. And in the subcutaneous white adipose of these patients, they reported increased gene expression of ATGL, HSL, and UCP1 in one or both of those weight-losing groups compared to the weight-stable cancer patients. Although it's still not entirely determined whether white adipose browning is actually occurring in human patients with cancer and if that's actually detectable at the whole body level and whether these pathways are really related to or associated with cancer-related weight loss. So our aim was to characterize the relationship between these pathways, both in the circulation and in white adipose tissue, in cancer patients with or without weight loss and in weight-stable non-cancer control patients. And we hypothesized that cancer-related weight loss would be associated with elevated energy expenditure in addition to being associated with markers from those 3 pathways. All the patients that we recruited for here at the Seattle VA, they were all males. Our cancer groups were either gastrointestinal or genitourinary cancer. These patients were all recruited either from surgical oncology or urology clinics that were planning an open abdominal surgery. And we obtained their pre-surgical clinically derived CT scans to quantitate adipose area. And then within a week or 2 before their surgery, we obtained a blood draw and performed indirect calorimetry to obtain resting energy expenditure and respiratory quotient. And then during the surgery, we got a sample of subcutaneous white adipose for gene and protein expression. So our 3 groups here were primarily on average in their mid-60s, mostly white, non-Hispanic. The cancer weight loss group had reduced body weight and BMI and elevated recent weight loss. There was no difference in those 2 cancer groups in the proportion of cancer types or stage or recent chemotherapy or radiation exposure. The weight-stable cancer group actually displayed the highest subcutaneous, visceral, and of course, total adipose area, while there was a trend for reduced respiratory quotient in the weight loss group, although not quite significant. And so I want to just point out really quickly, respiratory quotient is a measure of the ratio of substrates that your body is using for fuel. And you can see right around here is where the weight loss group is. They're essentially using about 1/3 carbohydrates and 2/3 fat for their energy usage at rest or during the fasting state. And the 2 weight stable groups are up here closer to 0.9, essentially using the opposite proportion of carbohydrates and fat. So we thought that might be an indication of some metabolic dysregulation happening in the weight loss group, especially considering we did not see any difference in absolute energy expenditure despite the weight loss group having a lower body weight. You would expect them to have lower energy expenditure, but that was not necessarily reflected, even though numerically that group has the highest relative energy expenditure. This was not significantly different across groups. In the circulation, we observed elevated IL-6 in the weight loss group with no difference in TNF, free fatty acids or PTHRP. And in the white adipose tissue, we observed elevated IL-6 and IL-1 beta gene expression in the weight loss group, elevated ATGL in the weight loss group, and elevated PGC1 and DIO2 in the weight loss group, which are upstream of UCP1, although we did not see any difference in UCP1, and strangely or unexpectedly, we saw an elevation in PTHRP gene expression in the control group. We also used immunohistochemistry to stain for UCP1, but did not observe any difference between groups. And then it's hard to do a lot of protein expression in adipose, but we were able to measure a few inflammatory related proteins, and again, did not see any difference between the groups. So this study has a few strengths of note, including the gold standard fat mass assessment by CT, and comparison of age-matched non-cancer control patients, and inclusion of both systemic and peripheral pathways. But we are limited by the lack of comparison between different adipose depots like visceral fat, and our lack of female patients, and our small number of protein expression. And then our cross-sectional design also limits our ability to infer any causation. But we did observe that cancer-related weight loss was associated with elevations in some white adipose, inflammatory, lipolytic, and browning markers, at least at the transcript level. And increased inflammation was confirmed systemically in the weight loss group, but elevated lipolysis and browning were not detected at the whole body level. And the presence of white adipose browning was probably unlikely due to the lack of alteration in UCP1 at the gene or protein level. So future studies should confirm these observations longitudinally to inform causative mechanisms, including the relevance of fatty acid oxidation and lipolysis and the lack thereof potentially for white adipose browning. Will need to be confirmed in other tumor types to help increase the generalizability of these results. So if anyone has the time to potentially scan this QR code and give a really brief review for my departmental peer reviews, I'd greatly appreciate anyone who can take the opportunity to do that. But of course, I have to mention the wonderful funding mechanisms and institutional resources and individuals that have made this project possible and made it possible for me to be involved in this project and complete this work, and including my mentor, Jose Garcia, and everyone associated with his lab, including Dr. Yiping Li at University of Texas Health Science Center, and our surgeons and our wonderful veterans who, of course, are the reason that we do what we do and help make all of this possible. Thank you very much.
Thank you, Lindsay. Great talk, important data for the field. As the questions come in, I guess I'll start with a question. You noticed that you measured resting energy expenditure, and given that cachectic patients probably have a reduced physical activity, wondering what your thoughts are on total energy expenditure and whether there would be any differences in that.
Yes, that's a great question, and that's hard to say because it's also very difficult to capture accurately anyways. And we do use some accelerometry in a number of our other protocols, and so we are kind of trying to look into that data in other cohorts, but I probably think that there's probably too much variability in the actual accuracy of capturing that method to make too many determinations. But I would say that probably, the weight stable groups actually tend to be much more overweight though than the weight loss groups. And so I think that their inactivity may come from different reasons. And that also may be a big factor in how that actually translates to over total energy expenditure.
I could continue going with questions as they keep coming in. We often see UCP1 message upregulated in the adipose tissue in animals with cachexia, but then have trouble identifying the protein on Western blot, for example. Wondering if you guys have tried maybe looking at fixed tissue for immunohistochemical markers of UCP1.
We did stain slides for UCP1 and did not. There was too much variability, really. The average was sort of numerically the same in the two weight stable groups and was about twice as high in the weight loss group, but too much variability to be statistically significant. So I definitely think there is some, too much. Either I'm not sure why, if there's instability or we don't have good good if it's the staining or what it is, but that's causing that variation. But I definitely think that is a big problem. That appears to translate from animals to humans as well.
Mm-hmm. We got a question from Teresa Zimmers. Wonderful study, Dr. Anderson. Have you tried to correlate the various molecular endpoints to the phenotypic endpoints on a per patient level? This analysis may reveal relationships that are masked by looking only at the group-wise comparisons. And similarly, the molecular endpoints with each other.
Yes, we actually are. I didn't have time to include all of that data today, but we do have a manuscript in review where we are showing the correlations between the systemic and the tissue levels, and actually finding very sparse correlations between those two. And so whether it's for lipolysis or inflammation, we don't I don't think PTHRP is actually a great systemic measure of browning per se. So I can't necessarily speak to whether there's a good, marker in the circulation, but I'm surprisingly finding that those are really not correlated with each other. And that kind of just confirms to me that those need to be, if you want to talk about tissue level mechanisms, you need to measure it there as well. And we have done some regression analyses with using all of these systemic and tissue markers, and including tumor type and BMI and weight loss, energy expenditure. And the thing that I'm finding to be the most predictive of, for example, adipose area is simply just BMI and not any of the molecular markers that we've measured to this point. So, so that's, kind of telling, I think, that BMI may be more influential than any of these pathways, possibly.
Right. And we have time for one more question from Maria Seelander. Excellent results. Have you seen the studies on UCP1 independent browning, and can that be happening? I believe she's referring to like calcium cycling or creatine cycling that have been shown in animal models.
We haven't looked into that ourselves, but we are aware that is that UCP1, there is browning independent of UCP1, and we are interested in whether that's something that is also happening. We haven't really started any of those things, but yeah, I'm kind of hopeful that actually, might be informative too, maybe more informative than UCP1.
Thank you very much, Dr. Anderson. I'll hand it over to Melissa for our next speaker.
My pleasure to introduce our next speaker, Dr. Shinpei Kuoka from Kyoto University, Japan. He'll be presenting on remote solid cancers rewire hepatic nitrogen metabolism via host nicotinamide and methyltransferase.
Hi everyone, I'm Shinpei Kuoka from Kyoto University, Japan. First, I want to thank the society and organizers for giving me this opportunity. This is actually my very first time to give a talk at this society. I'm so excited. Today, I'm going to talk about a novel host factor that is involved in cancer-induced abnormalities in liver metabolism. We consider cancer cachexia as a cluster of abnormalities in various host organs and cells. We think that comprehensive characterization of such abnormalities will help us to understand the nature of cancer cachexia. Our group is focusing on the entire picture of host abnormalities, responsible cancer and host factors, hierarchy of host abnormalities, and the direct causes of organismal death. To achieve these aims, we use a kind of simple strategy combining multiomics profiling and genetics. Here, let's take the liver as an example. Solid cancers remotely affect the multiomics network in the liver over time. At an earlier time point, perturbation should be limited to particular molecules, which I colored red in this picture. This abnormality eventually affects other molecules, and in the end, the entire structure of this multiomics network is rewired. Simply speaking, we comprehensively characterize these changes using multiomics analysis and use those datasets to find a lead factor. Identification of such factors makes it possible to understand the mechanisms of cancer cachexia and hopefully development of therapies that ameliorate cancer's adverse effects on the host. We recently found such an important host factor, nicotinamide N-methyltransferase, NNMT. NNMT is an enzyme highly expressed in the liver and adipose tissues. And this enzyme transfers a methyl group from S-adenosylmethionine into nicotinamide to produce methylnicotinamide, or simply MNAM. The function of MNAM is not clear to date, and it has been also unknown whether NNMT is involved in host pathophysiology in cancers, including cancer cachexia. Using transcriptome analysis, we found that multiple solid cancers increase expression of NNMT in the liver. In this experiment, we transplanted mouse cancers to mice of the same strain and analyzed the liver. As shown here, breast, colon, lung, and ovarian cancers all increased expression of NNMT in the liver in varying degrees. This was actually accompanied by the increase of the product of NNMT MNAM, as determined by the LC-MS analysis. Thus, cancers upregulated NNMT and MNAM in the liver. We then wanted to know the significance of this alteration. To address this, we generated NNMT knockout mice. We first found that deletion of NNMT completely abolished MNAM in the liver. This nicely validates NNMT knockouts and suggests that MNAM is produced only by NNMT. We then performed metabolome analysis against the livers of control and cancer-bearing mice, both for wild-type and NNMT knockout mice. Among 170 measured metabolites, a set of metabolites was going up and down by cancers. We then noted that NNMT knockouts suppressed part of these changes, especially upregulation of metabolites. Here I show you a heat map of metabolites that were significantly affected by cancers in wild type, but not in NNMT knockout. NNMT knockout buffered the increases of 36 metabolites in the liver. Looking closely into these datasets, we found that NNMT is involved in cancer-induced abnormalities in amino acid metabolism, urea cycle, and uracil biogenesis. Here are data for representative amino acids. Lysine and valine were accumulated in the livers of cancer-bearing animals, which were nicely rescued by NNMT knockout. From this data, we speculated that cancers disrupt amino acid metabolism via host NNMT. Consistently, we found dysfunction of the urea cycle, the very important pathway related to amino acid metabolism. The urea cycle is responsible for converting nitrogen-containing ammonia, which is in part produced from amino acid metabolism, into urea, which is non-toxic. In other words, the urea cycle is a nitrogen disposal pathway. From our metabolome data, we found that the livers of cancer-bearing animals accumulated a series of urea cycle metabolites, such as citrulline. In addition, plasma urea was reduced in the presence of cancers. These data suggest that cancers somehow suppress the urea cycle in the liver. The key in this slide is that NNMT knockout nicely buffered urea cycle dysfunction both at the liver and plasma levels. Thus, we concluded that cancers suppress the urea cycle via host NNMT. Interestingly, there is a trade-off between the urea cycle-dependent nitrogen disposal and uracil biogenesis. The key is aspartate, which is used for both pathways. Aspartate in one hand fuels the urea cycle, and in another hand used for biosynthesis of uracil that cells utilize for gene expression. Thus, more uracil biogenesis, less nitrogen disposal, and vice versa. Cancers appeared affecting this interesting metabolic link, increasing the amount of uracil, which was also rescued by an NNMT knockout. These metabolic changes are likely to be attributed to expression of CAD. CAD is the enzyme that is involved in a series of reactions that produce uracil from aspartate. Cancers increase expression of CAD, possibly enhancing biogenesis of uracil, which is again rescued by an NNMT knockout. Based on these results, we concluded that cancers suppress the urea cycle while enhance the biogenesis of uracil via NNMT. This is actually the first study that demonstrates a link among the NNMT pathway, urea cycle, and uracil biogenesis. Importantly, NNMT knockout buffered systemic phenotypes, weight loss, and the decrease for wheel running activity. On day 14 after transplantation of 41 breast cancer cells, wild-type mice lost approximately 2 grams of weight in our hand, which was at least in part rescued by an NNMT knockout. In addition, cancers reduced voluntary wheel running activity over time, which was also rescued by an NNMT knockout. At this point, however, causal relationships among these systemic phenotypes and metabolic changes in the liver remain unclear. In summary, we find that solid cancers increase expression of NNMT and its product MNAM in the liver, which in turn suppresses the urea cycle while enhancing uracil biogenesis. We also find abnormalities that do not require NNMT via multiomics analysis, such as inflammation and reduced glucose metabolism. We are on the way to identify other host factors responsible for such NNMT-independent abnormalities. For this purpose and thorough understandings of cancer cachexia, we are using these various measurements such as single-cell analysis, spatial transcriptomics, transcriptome, metabolome, proteome, etc., in combination with mouse genetics and human samples. We are, just as an introduction, we are basically a team of omics measurements and bioinformatics. A lot of data sets are accumulating in the lab, and I hope this meeting will give us a chance to have a new collaboration with people in this society. Finally, I want to thank everybody involved in this project. So thank you very much for your attention. I'm now happy to take any questions.
Thank you, Shinpei. Great study. We have a couple of questions coming in the chat. The first one from Teresa Zimmers. Very nice study, well-powered and robust. Could you please clarify the models? Was it liver-specific NMNT knockout and the route of 4T1 administration location? Does the 4T1 metastasize to the liver in your model?
So it's whole body knockout, but we have an NMT knockout both for BRCA6 and valve-C, and we use cancer models using both strains. And we have single-cell transcriptome data in the livers of 41-bearing animals, and we don't see any small metastasis to the liver on the time point we are using.
Okay. Our next question, is there any?
Oh yeah, sorry. I forgot answering one question. So root of 41 administration, it's in the fat, a mammary fat.
Is there any association between higher expression of NMNT in livers and abnormal liver enzymes like AST?
So I've never seen association of the degree of NMNT expression and other liver enzymes like AST.
This one is from Marcus. Do you have any plans for flux analysis in your mice to trace nitrogen?
So it's really important question. We want to do that experiment, but we haven't done yet. And we need some collaborators to do that experiment.
Have you observed any change in NAD levels in your metabolite or metabolomics analysis?
So yes, cancer transplantation massively deplete NAD from the liver, which was at least in part a little bit rescued by an NMT knockout.
Do you think cancer use any of the upregulated metabolites for its benefit?
Yeah, I kind of hope so and think so. And for example, we detect the increase of uracil even in the plasma. So it's in the blood flow. So I guess cancers may use those metabolites produced by host, or they force the host to produce metabolites that are useful for their proliferation. But this is just a guess.
As a follow-up to Dr. Zimmer's question, would you expect exacerbated upregulation of NMNT with liver mets?
Actually, I don't know. It's interesting if this is the case.
Was the T4 tumor progression comparable in the NMNT knockout and wild type mice? Does NMNT knockout in the host affect the T4 tumor progression?
So actually the size of 41 tumors are comparable between wild type and NMNT knockout. So it looks like NMNT knockout does not affect 41 tumor progression. But I have one opinion for this comparison. So we inject lots of cancer cells at once to the host. So it's so cancers can proliferate so much even without increase of NMNT. So because this is artificial experimental setting, this is my guess.
Okay. Fantastic talk. I'll turn it back over to Marcus to introduce our next speaker.
All right, our next speaker is Young-Mi Kim from the University of Illinois at Chicago. Downregulation of PGC1 alpha in the skeletal muscle endothelium mediates cancer cachexia by compromising vascular barrier integrity. Take it away, Dr. Kim.
Okay. Thank you for introduction. Hello, everyone. My name is Young-Mi Kim from University of Illinois. Today, my talk title is Downregulation of PGC1 alpha in the skeletal muscle endothelium mediates cancer cachexia by compromising vascular function. What is the relationship cancer cachexia and endothelial cells? Over 80% of advanced cancer patients experience cancer cachexia, and loss of muscle mass strongly contribute to an increase in motility. And our body, blood vessel lined by endothelial cells, regulate the function of all organs by supplying nutrients, oxygen, and signaling cues. However, the role of muscle endothelial cells in cancer cachexia is not well understood. Recent papers suggest muscle satellite cells crossed with the vascular niche to maintain their sameness, and endothelial lactate controls muscle regeneration from ischemia by regulating macrophage polarization. Moreover, several studies in other organs suggest that restoring endothelial health and also reverse tumor dysfunction. So this opens up the possibility of targeting muscle endothelial health to improve muscle functions. So we had a question, do tumors induce muscle endothelial dysfunction and thus compromise muscle functions? To address our question, first we use tumor allograft mouse models. Mouse skin melanoma cells transplanted subcutaneously at right front region, and time-dependent manner we followed the cancer Cachexia marker expression. And around 3 weeks later, we found cancer, one of the key cancer Cachexia markers, E3 ubiquitin ligase, morphone and atrogin 1 significantly increase in skeletal muscles of melanoma-bearing mice. These mice also significantly decrease mouse forelimb grip strength. So how about muscle vascular density? And we use skeletal cross-section to stand muscle endothelial cells with CD31 antibody. As you can see here, wild-type mice skeletal muscles beautifully show high density of CD31 positive vasculature. However, melanoma-bearing mice markedly decrease CD31 positive density, indicating this localized tumor remotely controls skeletal muscle vasculature. So we like to confirm our observation with different mouse models. So we use KPC mice because KPC mice mimic human pancreatic ductal adenocarcinoma, and to produce KPC mice, we cross KRAS mutant and p53 mutant mice with pancreatic-specific promoter PDX1 mice. These mice gradually form the tumor in pancreas. Around 5 months later, we found cancer Cachexia marker significantly increase in skeletal muscles. That's why we defined these KPC mice to KPC Cachexia. How about muscle vasculature? And we took the advantage of innovative three-dimensional tissue imaging system, and wild-type mice show high density of muscle vasculature and show they formed a very good network. However, cancer Cachexia mice markedly decrease muscle vascular density and show broken networks. So we wonder how about the human muscles. Surprisingly, cancer patients show severe muscle atrophies. Under this condition, we found cancer patients also significantly decrease human endothelial cell marker UA positive endothelial cells. So based on our result strongly suggests reduced vascular endothelial density, one of the key indicators for cancer Cachexia. So next question was, what are the consequences of the decreased vascular density and cancer Cachexia muscles? To address our question, we performed vessel permeability assay using gypsy conjugated albumin. All mice injected with gypsy conjugated albumin through vein and perfused with PBS. So if mouse have the intact vessels, almost albumin will be washed out. However, if mouse have the weak vessel or leaky vessel, albumin diffuse into the surrounding tissues. As you can see here, melanoma-bearing mice highly increase green fluorescence, indicating a fixed conjugated albumin into the skeletal muscles, indicating melanoma-bearing mice increase muscle capillary leakage. Under this condition, we found several pro-inflammatory cytokine gene expression level also increase. So we like to understand how happened this kind of phenomenon. And we performed the bulk RNA-seq analysis using muscle vascular endothelial cells from tumor-bearing mice, and we did unbiased analysis. And here we present top 10 gene ontology analysis. Surprisingly, we found muscle endothelial cells significantly increase muscle cell differentiation related genes as well as loose metabolic process related genes. However, catabolic process or developmental growth related gene was significantly decreased. What's the meaning? We found under the Cachexia condition, muscle vasculature is decreased. However, survivor remaining endothelial cells try to compensate their functions. If we look at the more detailed endothelial loss metabolism related genes, we can see peroxyreduction related family gene GPX1 SOX3 expression level highly increased during tumor formation. tumor growth, indicating reduced muscle vasculature, induce hypoxia, and increase loss formation. So endothelial cells need to increase this kind of antioxidant enzymes to compensate. The next question was, can we restore muscle endothelial health and muscle function in cancer cachexia muscles? Interestingly, we found skeletal muscle endothelial cells from melanoma-bearing mice as well as KPC cachexia mice. Endothelial PGC-1 alpha expression level was markedly decreased. As you know, PGC-1 alpha is transcriptional cofactor, is one of the key regulators for mitochondrial biogenesis for oxidative tissues such as heart, liver, muscles. And PGC-1 alpha attenuates cell death and increases cell proliferation. However, the role of PGC-1 alpha in endothelial cell is not fully understood. So based on this result, we thought if we overexpress endothelial PGC-1 alpha into the muscle vasculature, we may could restore muscle functions in cachexia condition. So we developed a lentivirus expressing endothelial specific PGC-1 alpha conjugate with GFP. So here we visualize endothelial cell with another endothelial marker isolating before with red color. And tumor-bearing mice significantly decrease vascular density. However, indeed, endothelial PGC-1 alpha overexpressed muscles clearly rescued the vascular density as well as cross-sectional area. Also, grip strength also clearly rescued. So based on our result, we made the conclusion under the tumor-bearing condition, muscle endothelial cells decrease endothelial PGC-1 alpha levels and induce its loss and increase leaky vessel. That's why contribute muscle loss and cachexia. If we overexpress the endothelial PGC-1 alpha into the muscle vasculature, we could increase healthy endothelial cells and normalize vessel function and increase muscle mass and reduce cachexia. So we like to propose endothelial PGC-1 alpha is required for maintaining mass and strength of skeletal muscles, and restoring muscle endothelial dysfunction could be a novel therapeutic approach to prevent or reverse cancer cachexia. So I would like to thank you, my mentor, Dr. Charles Riemann, and our lab board member, especially Sarah Bianca. And for the imaging, I got a lot of help from RRC imaging coordinator, Dr. Peter Toff, and 3D tissue imaging by Steve Lee, and KPC mice supported by Dr. Paul Grippo and George, and human muscle sample supported by UI Cancer Center, and pathologist Dr. Tiba Barinegi evaluate human samples. Here is our funding source. Thank you, everybody. I'd like to take your question.
Thank you very much. Great talk. Very interesting new look on what could be happening to the muscle almost indirectly. Question in the chat. In your KPC mice, do you know what time point during the tumor cachexia progression that skeletal muscle endothelial cells are reduced? Does this reduction precede the loss in muscle and myofiber atrophy?
Yes, that is a great question. We also had the same question. That's why we look at the time course. And we found around 5 months later, after born, we found the cachexia marker gene increase as well as body loss, loss of muscle mass. However, we could see muscle vascular endothelial density start to lose from the 3 months.
Very nice.
Yes.
Fabio Penna has a question. Great talk. Have you measured PGC-1 alpha expression in downstream target genes in other muscle microenvironments?
Great question. We are focusing in endothelial cells and non-muscles. So we look at the PGC-1 alpha downstream gene in endothelial cells. And today I didn't present my old data, but as you remember my in vivo data, cachexia muscle highly increased capillary leakage. And then we found endothelial barrier function is disrupted. So we found PGC-1 alpha regulate expression of one of the endothelial adherent junctional protein, V-cadherin.
So this is my own personal question. Do you think
Yes.
that the endothelium are normally filtering out something that would not be present in the muscle, but then in the presence of cachexia, it leaks into the muscle?
We think endothelial cells should tightly control any kind of transmission by controlling their barrier. But under the cachexia condition, endothelial cell density is decreased. That's why their barrier function also lose. And then with this kind of environment, probably several inflammatory cytokine and inflammatory cells infiltrated into the muscle tissues. We are thinking.
Yes, additional question from the chat. Do you think that aerobic exercise may help the endothelial cells and protect them?
Of course, but I think that from the Friday, we always thought cancer patients very difficult to exercise. So we don't know how we encourage too much to the exercise, but of course, I guess probably will be helpful.
Yes, next question. Did you study any impact of this endothelial dysfunction with muscle regeneration markers?
Very good question. We already start. We knew endothelials are very much important to regenerating muscles. So we have already preliminary data. Endothelials are actually already 2018 published cell metabolism paper. They also proposed endothelial cell regulate stem cell regeneration. So we also could found cachexia muscles markedly decrease muscle satellite cells. That is correlated with vascular density. So, and then we also have the rescue effect. When we overexpress the EC-specific PGC-1 alpha, we could increase muscle satellite cells.
Great. Question from Dr. Bonetto. Very nice talk. Out of curiosity, is it known if vascular density normally correlates with muscle size? For example, larger muscles are more vascularized, whereas smaller muscles have fewer vessels.
Yeah, great question. We also like to check the different muscle type and the muscle size, but so far we haven't done that much detail. But we know muscle size also affects.
Mm-hmm. And two more questions. Question from Xiaoling Zhang. Are post-translational modifications of PGC-1 alpha, phosphorylation, for example, involved in this process?
Great question. One of my interesting also, I like to look at the post-translational modification. So far, what we found before post-translational modification, we already found cachexia muscle already decreased PGC-1 alpha expression level in RNA and protein levels. So that's why first protein level, gene level, as well as protein level itself also more important. Later probably is very much worth to look at the PGC-1 alpha itself modification. But first we like to look at how gene level as well as protein level of how affect muscle functions in endothelium.
Yes. And last question from Josh Huat. Curious if there are plans to assess if chemotherapy has similar effects on the endothelial cells, given that cachexia inducing effects of anti-cancer drugs.
Good question. So far, we have been trying to combine with chemotherapy or radiotherapy, but we have the plan. We like to combine with some nutrient supplements together and how they help to increase vessel healthiness. So let's see. Still, we couldn't get approval from the animal protocol. So yeah, let's see.
All right, thank you very much for a great talk and good discussion. I'll hand it over to Melissa.
Our next speaker is Dr. Fabrizio Pin from Indiana University School of Medicine, and he'll be presenting on mitochondria-targeted agent MitoQ improves muscle atrophy, weakness, and oxidative metabolism in C26 tumor-bearing mice.
Hello, everyone. It's a pleasure to be here. And thank you, the organizer, for the opportunity to give me to show some of our data today. So yes, so as you can see from the title, I will present the possibility to use an antioxidant targeting mitochondria in MitoQ to improve the cachectic phenotype in preclinical model. Well, I don't need to introduce cachexia. I just want to say that is an energy syndrome, and in particular, there is an energy negative balance that's obtained, this alteration. Mitochondrial dysfunction contribute to this energetic negative balance. A lot of work are due to understand and clarify mitochondrial structural damage, alteration of the oxidative metabolism, shift from oxidative to glycolytic metabolism, reducing of ATP production, ROS that can damage the mitochondria. All of these can contribute to this energy negative balance. What we can do to improve that? Well, we have different strategy. We can use, for example, exercise, physical exercise in different modality. We can use exercise mimetic. We can use metabolic modulator. We can use antioxidants to protect the mitochondria for the damage. And all of these is to maintain the quality of the muscle that is fundamental for the homeostasis, and not only just the mass and the function, but also the metabolic level. We are focusing today on the mitochondria target antioxidant. So this, you can see there is, for example, MitoQ, SKQ1, SS31. These molecules go inside of the mitochondria and they're acting, reducing the reactive oxygen species. We know that they are able to reduce insulin sensitivity, for example, have an effect in the age-related decline. Also, there is some information that is not really clear about their function in the mitochondrial biogenesis and the contraction. Some months ago, Fabio Penna's group showed this paper, published this paper, sorry, when they showed that the SS31, this is one of these compounds, was able to improve the overall metabolism in the cancer and chemotherapy-induced cachexia. What we choose to do is use the MitoQ. MitoQ is a molecule that the active part is a ubiquinol that is oxidated in ubiquinol and then reducing ubiquinol. So this is the active part. This MitoQ, this active part is conjugated with an cation. You can see here the name is triphenylphosphonium. This molecule is able to accumulate in the biological membrane thanks to this cation, and in particular they are accumulating 100 times more in the mitochondrial membrane due to the different potential that there is in this membrane. So the aim of this study was to testing the effect of MitoQ administration in improving body wasting, muscle atrophy, and weakness in the C26 tumor-bearing mice. So for answering this question, this is the experimental design. I was just add something that we had that in the slide before. The MitoQ is something that is available. You can buy in Amazon. So basically, it's used for the people by like a supplement. So to design this experiment, we decide to make a long pre-treatment. You can see that we start 3 weeks before the injection of the tumor to treat the mice with the MitoQ until the end of the sacrifice. These mice were treated with MitoQ in the water ad libitum, and we divide our experiment in 4 groups: healthy mice receiving MitoQ or the triphenylphosphonium, that is the cation, the inactive molecule, and then C26, the mice treated with MitoQ and obviously with triphenylphosphonium. So we start with some data. You can see that the C26 model is doing what to do. The mice, they start to losing weight after 8 days, and they're really cachectic at the end of the experimental period. The MitoQ was able to partially a little improve the body weight. You see also here by the difference from the final body weight and initial body weight. This is the body composition analyzed with the echo MRI. You can see there was a little improvement of the lean mass, not too much in the fat mass, all without changing the tumor size. We decided to go directly and see what the MitoQ was doing in gene related with the mitochondria. So we analyzed some gene of biogenesis. PICC1 alpha was changing, mitophagy, Parkin, and PINK1 was not changing. Dynamics also was some difference in the mitofusing one. We saw some difference in the gene related with the function, like cytochrome B, also cytochrome C with the t-test, but with the ANOVA is not more significant. So we decided to go more deeply and make more functional metabolic analysis. And here, some of these alteration was already described before, and in particular in the muscle of the C26 tumor-bearing mice, we observe an altered glucose metabolism. In this case, we observe an increase of exokinases. This is really important and key enzyme of the glycolytic pathway. We saw an increase of PDK4s. We already published that PDK4 actually is really upregulating a more different model of cancer cachexia. And also in this case was increased. PDK4 is a kinase that is able to inhibit the function of PDH, and PDH indeed was reduced in the activity in the muscle of our mice, as well as the succinate dehydrogenase, another enzyme of the TCA cycle. They also have an important role in the electron change. The treatment, what did was able to reduce the activity of the exokinases, reduce the expression of PDK4, so allowed more activity for PDH. Indeed, the pyruvate dehydrogenase activity was increased, as well as increased the SDH activity. So, in the end, the oxidative to glycolytic shift is corrected by the mitochondria. Another alteration that we a lot of time we saw in the muscle of our tumor-bearing mice is the accumulation of fat in the myofiber. So after we published, we saw that the C26 tumor-bearing mice, we have a high level of LDL in the circulation. And we already showed that the LDL receptor is hyperexpressed in the skeletal muscle of the C26. Here again, we show that the receptor is hyperexpressed. And also we show that there is an accumulation of fat inside of the fiber. This is the oil red oil staining. The treatment didn't change the expression of the LDL receptor in the muscle. So this means that the uptake of the fat or the LDL is the same also with the treatment. But we observe a reduction of the staining for the oil red oil. So this means that the fat is updated the same, but probably with the better metabolism, the muscle is able to process the fat and not be accumulated. An indication of that was also due by the increase of the hormone-sensitive lipases, that is an enzyme that is inducing lipolysis that was increased by the treatment. This is in the muscle, so they say that lipolysis is acting in the skeletal muscle. So this change in the glucose metabolism, in the TCA cycle, improvement of the oxidative metabolism, this change in the reduction in the fat accumulation were associated with clearly phenotype. There was a rescue of the partially rescue of the skeletal muscle mass. You can see here in the tibialis mass, in the gastrocnemius mass, and also this increase of the mass was consistent with an improvement of the strength. This is measured by the grip strength test. This morphological and functional data was also consistent with a partially correction of the expression of important markers of protein catabolism that normally are upregulated in the skeletal muscle of the tumor-bearing mice. At the end, we also started to perform some in vitro experiment. In particular, here are C12 myotubes exposed to the condition media of C26 that is able to induce myotubes atrophies. We make a co-treatment with mitocure 75 and 150 nanomolar. The treatment at both concentration was able to improve myotube size. So we decided for the molecular characterization, we use the lower dose with the same effect. You can see that also in vitro, we proposed that the increase of the marker of catabolism was partially corrected by the treatment. Also, the alteration of the metabolism due to increase of PDK4 was partially corrected by the mitocure administration, as well as the hormone-sensitive lipases that is reduced, maybe suggesting that also in this in vitro there is accumulation of fat, was pushed by the mitocure. More we saw that there is some indication that the catabolism is inhibited by probably by the mitochondria. We want to check also the reverse if what happened to the anabolism. So we have started to check two markers, and in particular the ratio between phospho-AKT and AKT. It was really an increase. You can see also from the picture here of the increase of the phosphorylation, also in the healthy animals just without the tumor. And this was also consistent with a regulation of the ratio between the phosphorylation of 4-EBP1 and EBP1. That is an important for the protein synthesis. So we have some indication that also probably the anabolism is improved by the treatment. Obviously, more analysis we need to do to better understand in vivo. This is in vitro. Whatever. So to summarize our data, in the skeletal muscle of the tumor-bearing mice, we observe an altered metabolism, increase glycolysis, reduction of the TCA cycle, and increase of myosteatosis, accumulation of the fat in the muscle. The mitochondria is able to reverse the oxygen to glycolytic metabolism, I show here, and also is reducing the myocytosis. So we are speculated that the product of the lipolysis at this point, because the metabolism, the TCA cycle is working better, if they are able to be processed and used as a fuel to produce energy. So basically, in conclusion, our data identify mitochondria as an effective treatment to improve skeletal muscle function in tumor host and support studies aiming to testing anti-cachectic property of mitochondria in combination with routinely administered chemotherapy agent. And I would like to thank you, my mentors, Andrea Bonetto and Linda Wodewald, and in particular also Joshua Hua, that is my colleague, and we share the bench every day, and also Rafael Barreto. And thank you, all of you, for your attention. Thank you.
Thank you, Dr. Penn. Wonderful talk. We have a whole bunch of questions coming in. So the first one is from Marcus, and he says, I see that mitochondria has a long carbon chain. Is this a nutrient? Would a similar length lysophospholipid have similar results?
Well, this is an interesting question, and well, I don't have a response. I don't have the idea, but it's possible.
Our next question is, have you had the possibility to measure energy expenditure in cachectic and mitochondria treated mice?
No, we didn't have the chance, but I think that with something that we do for sure. I think that something that we will do for sure is, for example, that we have some more functional, starting with more functional experiment, mostly in vitro, also making some analysis in the seahorse to see really there is a difference in the respiration.
Got time for a few more questions. Did your C26 groups alter food intake? If so, were your non-tumor groups pair-fed to the C26? Would food intake per se play a role under mitochondria treatment?
Well, this is a very interesting question. Unfortunately, we didn't measure the food intake. Normally, we keep all the mice in the 5 mice per cage, so also we didn't have the possibility to measure. But I think that it's something that if we need to clarify better at this point, is something that we will need to do is look in the diet. Yeah.
Okay. Have you looked at tissues other than muscle, like the heart, the liver, or the tumor itself?
Yes, well, the tumor itself, just the mass is not changing, but some suggestion that I spoke yesterday with Maria Slender, she told me that would be possible also that maybe it's not change the mass of the tumor, but probably we need to check if there is maybe different necrosis inside, so there is changing in the composition of the tumor. We checked the heart, yes, we didn't see any difference, as well as we didn't see any difference in the liver. We saw difference that I didn't put, sorry, here, because we're just more focused on the muscle in the white adipose tissue. There was, we know this really reduced in this model, but the treatment actually was able to increase substantially the amount of fat. Now we need to be more clear and understand why this, but yeah, for example, we saw that.
And we'll do one more question. what's the outlook for the clinical use of MitoQ in patients with cancer cachexia, given that it's readily available and must be generally regarded as safe in normal people?
Yes, is available. Yes, is safe, but I don't think that is FDA approved. Really, the people is just thinking like a supplement, and I think that will be nice to be tried to like use as a treatment. In this case, we use like a supplement, like a prevention. But what was nice is that also if we pre-treat the mice for 3 weeks at the baseline, when we measure the strength from the baseline, there was no difference. So this means that maybe it's not really acting in physiological conditions. So the mice are starting. The MitoQ was not in a better or better condition with respect to the treated one. So I think that can be speculated. We can try at least to use like a treatment, not only like we show here, like prevention.
Great talk. I think that's all the time we have for questions. There's a few more in the question box if you want to answer those. And with that, I will turn it over to Marcus to introduce our next speaker.
Thank you, Melissa. Our next speaker is Joseph Rupert from Indiana University School of Medicine, and his title is his talk is titled "Deletion of PKC theta in mice with PDAC is associated with increased survival, increased in vivo muscle force production, decreased muscle wasting, and reduced expression of PDK4 in skeletal muscle." Take it away, Dr. Rupert.
Thank you for the introduction. Thank you all for attending today. And as was stated, I'm going to talk a little bit about my work looking at the role of PKC theta in PDAC-associated cachexia. So just to start with a brief background and context of the project, I primarily work with pancreatic cancer cachexia. Pancreatic ductal adenocarcinoma, or PDAC, is associated with a severe cachectic phenotype. Approximately 85% of patients with PDAC will be diagnosed with cachexia. Annually in the United States, there are about 60,000 new diagnoses of PDAC and approximately 48,000 deaths attributed to PDAC. Pancreatic cancer has one of the lowest 5-year survival rates across cancers. Currently, it's around 10%. And cachexia is important in pancreatic cancer because it significantly increases patient morbidity and mortality. And as we all know, cachexia is a multifactorial syndrome, and it's proven to be difficult to treat. Just to kind of explain how we started to look at PKC theta, we've recently published a paper where we looked at the crosstalk between the pancreatic tumor, adipose tissue, and skeletal muscle using an orthotopic model. We took isolated cells from the KPC mouse model of pancreatic cancer developed by the Tuvison laboratory, and then orthotopically implanted those into C57 black 6 mice. And one of the main findings from the paper was that IL-6 and IL-6 transsignaling between fat and muscle was important. But we also discovered that the crosstalk between adipose tissue and muscle was largely dependent on severe and chronic lipolysis, where as the tumor-induced adipose wasting, this increased fatty acids in circulation, and these fatty acids were accumulating in skeletal muscle as lipids, otherwise known as myosteatosis, and this significantly had a negative effect on muscle hypertrophy. And then here, just to take away from the paper, these are cross sections from quadriceps with either the no tumor mice or the KPC tumor bearing mice. And they were stained with oil red O, and oil red O identifies neutral lipids in tissue with a red color. So you can see here, compared to the no tumor mouse muscle, we see a significant increase in the accumulation of lipid droplets in the myofibers from the KPC tumor bearing mice. So based on these results, we asked the question, what lipid signaling pathways or lipid signaling molecules might be at play here in cancer cachexia, and more specifically muscle wasting. So just combing through the literature, we found lipid signaling to be important in insulin resistance and diabetes, as well as muscular dystrophy. And one common denominator that we found in these studies was the activation of protein kinase C theta, or PKC theta. And then again, another common denominator from these studies was that when they either inhibited PKC theta or ablated PKC theta in vivo, they saw a significant improvement in the muscle physiology in these disease models. So when we looked at PKC theta signaling, we found it interesting that a lot of the downstream pathways that PKC theta activates have been implicated in cancer cachexia. So PKC theta is unique in that it requires diacylglycerol to be present for its complete activation. Studies have shown that when fatty acids increase and muscle accumulates fatty acids, diacylglycerol goes up in skeletal muscle. And a few of the pathways that PKC theta can regulate that are involved in cachexia, PKC theta has inhibition of insulin signaling, so this would have a negative impact in skeletal muscle metabolism. It can also increase reactive oxygen species, and then it can also increase activation of the NF-kappa B pathway, which would increase inflammation and the production of cytokines. So when we looked at activation of PKC theta in our model, we found that, again, compared to muscle in the no tumor bearing group, we saw a significant increase in the phosphorylation of PKC theta in muscle from mice bearing KPC tumors. So we then went on to ask the question, what happens if we delete PKC theta in this model? What are the effects on cancer cachexia? So again, using the orthotopic model, KPC cells were injected either into wild type mice or PKC theta knockout mice. And then in collaboration with Andrea Bonetto's laboratory, and more specifically, this in vivo work was done by Joshua Hewitt, we looked at differences in vivo muscle force production using the Aurora Scientific force transducer platform. We looked at 2 time points, 8 days after tumor implantation and 15 days after tumor implantation. And what we found compared to the wild type mice with tumor indicated here by the black curve, we saw no difference in force production between the knockout mice with tumor in the red or the wild type mice with tumor in black. However, at day 15, we saw that the knockout tumor-bearing mice had significantly higher in vivo force production compared to that of the wild-type tumor-bearing mice. These mice were then euthanized, and we looked at differences in muscle wasting by tissue weight. Across 3 different skeletal muscles, we saw significant attenuation of muscle loss in the knockout tumor-bearing mice for quadriceps, gastric venous, and tibialis. And we also saw a sparing of cardiac wasting in the knockout tumor-bearing group. The reduction in skeletal muscle wasting was further supported when we looked at myofiber cross-sectional area. Again, these are cross-sections from the quadriceps reacted with dystrophin. And what we found was that wild-type tumor-bearing mice had a significant reduction in the average myofiber cross-sectional area. And this reduction was essentially rescued when PKC theta was deleted out of the mouse. We then went on to perform proteomics on the quadriceps muscles in these mice. We used ingenuity pathway analysis to analyze our proteomics data to look at different signaling pathways that were being regulated in the model. And what we found when we looked at the wild-type tumor-bearing mice, a number of pathways were affected. IL-6 turned out to be a central node in regulation and the overall changes in these pathways. However, we found a number of pathways that were related to lipid metabolism or lipid transport. We saw an increase in fatty acid metabolism, a decrease in glucose metabolism, and an increase in the transport of lipids, as well as the efflux of cholesterol. And then when we performed the same analysis in the knockout tumor-bearing mice, again, we saw IL-6 as a central node, but we saw the absence of the activation of those lipid signaling pathways. We took it a little bit further and we looked at the top 10 upregulated proteins and the top 10 downregulated proteins between each group. The top upregulated protein in the wild-type tumor-bearing group was pyruvate dehydrogenase kinase 4, or PDK4, as well as 2 other proteins here that are involved in lipid signaling and metabolism, ApoE and FABP4. When we did the same comparison in the knockout tumor-bearing mice, PDK4 didn't make the list of top regulated proteins, nor did those other proteins, ApoE or FABP4, involved in lipid metabolism and signaling. So just to confirm what we were seeing from the proteomics, we did Western blotting in the quadriceps from mice in these groups. Then we looked at PDK4 expression. So you can see here, the wild type tumor bearing group has a robust expression of PDK4 in their skeletal muscle. And then when PKC theta was deleted out of the mouse, we didn't see that increase in PDK4 expression. And that was quantified over here. And we find PDK4 to be an interesting target in pancreatic cancer. Previously, labs, especially Andrea Bonetto's lab, has published data indicating that PDK4 is a driver of metabolic alterations as well as muscle atrophy in models of cancer cachexia. So again, following the experimental design from the PKC theta experiment, we obtained PDK4 knockout mice. So these mice had to be cryo recovered, so they were heterozygous for the PDK4 deletion. So we had to breed them, obviously. So this is largely a pilot study at this point, and we're currently working on increasing our sample size. But the experimental outline here, we had wild type mice that were orthotopically implanted with KPC tumor cells. We had PDK4 heterozygous mice that were also implanted with the KPC tumor cells. And then we had PDK4 homozygous knockout mice, again, implanted with KPC cells. And then again, in collaboration with the Bonetto laboratory and Joshua Hewitt, we looked at in vivo muscle force. Here, this chart showing maximum muscle force in vivo at day 15. Again, we saw a significant reduction in the wild type tumor bearing mice for in vivo muscle force. And we did not see a significant reduction when PDK4 was deleted out of the mouse. Josh performed a fatigue analysis, which is essentially a series of rapid 15 contractions sequentially in vivo. And what we saw indicated here by the black curve was that wild type tumor bearing mice had a significant reduction in vivo force production as the contractions progressed. And then we didn't see that reduction in force production. We didn't see the reduction in force when PDK4 was deleted out of the mouse. And this was quantified by measuring the area under the curve and indicated over here by this chart. Again, the mice were euthanized and we looked at tissue weight as a readout for muscle wasting. Across the 3 skeletal muscles, again, quadriceps, gastrocnemius, and tibialis, we saw significant wasting in the wild type tumor bearing group. And then when PDK4 was deleted out of the mouse, in the tumor bearing group, we did not see significant muscle wasting. So deletion of PDK4 was associated with an attenuation of muscle atrophy, at least skeletal muscle atrophy. So one of the interesting results that we found, while it seemed that deleting PDK4 was protective in skeletal muscle, it seemed to have no effect on cardiac wasting. So across all 3 tumor groups, they had significant wasting of cardiac muscle. So then just to summarize and conclude what we found so far, PDAC cachexia is associated with myosteatosis. Deletion of PKC theta improved both in vivo muscle force production as well as attenuated skeletal muscle and cardiac wasting. Deletion of PKC theta was also associated with a reduction in PDK4 expression in skeletal muscle. And deletion of PDK4 attenuated skeletal muscle loss, but did not seem to be able to rescue cardiac wasting. So just to conclude right now, myosteatosis seems to be a promoter of muscle atrophy, as well as an activator of PKC theta in skeletal muscle. PKC theta from our data seems to be an upstream regulator of PDK4 activation. And we think that targeting either PKC theta or PDK4 may be a beneficial therapy for treating cancer cachexia, more specifically muscle wasting. And then with that, I'd just like to thank my mentor and PI, Teresa Zimmers, all of our collaborators and laboratory members, the Cancer Cachexia Society for putting on another great conference, and I couldn't include all the data in my talk here, but I do have a poster. It's number 58, and it has a lot more data pertaining to the project. And with that, I will take any questions.
Thank you very much. Great talk. We have a few minutes for questions. Very nice talk. Is there a treatment to target PKC theta, possibly a small molecule?
Yes, there is. There are actually a number of small molecule inhibitors for PKC theta. Like I said, I wasn't able to include all the data, but we use a small molecule inhibitor called sotrastorin to treat myotubes that were also co-cultured with media or plasma from tumor-bearing mice. And the PKC theta inhibitor, sotrastorin, seemed to rescue myotube atrophy in the presence of cachectic plasma.
Great. Very interesting talk. Have you looked at taking muscle from human patients with PDAC resections to see if the expression is also reproduced?
Yes. So we do have samples from a human cohort that we had a study here at IU for. We haven't been able to look at them recently. A lot of this is pretty recent data, but we do plan on taking those human muscles and sectioning them, and or even making lysates and looking at PKC theta and PDK4 expressions.
Thanks for your great presentation. Do you have any special reason to choose the muscles you chose, gastroc, tibialis, and quad in your study?
No, I guess not a specific reason. A lot of studies in the past have mainly looked at maybe tibialis or gastroc. I just kind of wanted to sample more muscles. I also have soleus that I would like to start to look at, as well as the diaphragm. So just kind of trying to expand out and just get as many skeletal muscles as I can look at.
I'll just add on there. Do you also see atrophy in the soleus in your mice?
So unfortunately I didn't measure tissue weight in the soleus. I just took tissue blocks so I could section them, and I haven't had the chance yet to look at them.
Maybe interesting to see a lack of PKC delta activity in those muscles that do not atrophy.
Right. Just to add on to that, one of the things that we think is happening is oxidative fibers are being preferentially affected against glycolytic fibers. So the soleus is really oxidative, so it'll be interesting to look at that as well.
Yeah. Okay, last question. Do you know if muscle regeneration was also improved in the PKC theta knockout when implanted with KPC, and also in the setting of PDK4 knockout?
So we have not looked at muscle regeneration yet in these models, but it would be interesting to see if that's affected or stem cell differentiation, those types of things.
Excellent. There's one more question in the chat that I'll let you answer regarding cardiac atrophy. Interesting question. Sorry, we don't have time to go into that. And I'll hand it over to Melissa for our last speaker.
Thank you, Marcus. Our last speaker is Walter van der Worp from Maastricht University in the Netherlands, and he'll be presenting on automated quantification of tumor volume and characterization of cachexia in a novel orthotopic lung cancer model.
Thank you. And I also would like to thank the organization for giving me the opportunity to present here my work. And my name is Wouter van der Worp, and I'm a PhD student from the Department of Pulmonology at the Maastricht University in the Netherlands. And today I will present you my work about the automated quantification of tumor volume and the characterization of cachexia in a novel orthotopic lung cancer mouse model. Oh. I'm not here, yes. So experimental animal models are still very important to increase our understanding on cancer cachexia and to evaluate the therapeutic effect of interventions. However, one of the greatest challenges we encounter as researchers is the translatability of these models. In lung cancer cachexia, the Lewis lung carcinoma mouse model is the most commonly used model at the moment. And in this model, the tumor cells are injected under the skin, and this results mainly in a relatively large ectopic growing tumor, which often represents up to 10% of the total body weight, and results in a rapid symptom onset. And this is not really what we see in the human patient. So in order to study lung cancer cachexia more realistically, orthotopic mouse models in which the tumor grows in its original stroma, where it can interact with the complex microenvironment, are needed. So if we then look at the table, we see that these orthotopic mouse models much more recapitulate the human disease. However, the injection of the tumor cells and the monitoring of the tumor development is much more complex and requires specific skills. So orthotopic mouse models are much more relevant, but we need to resolve some challenges. So as better models are needed, we started to develop a novel orthotopic single nodule mouse model to study lung cancer cachexia. And to create this model, I make a small incision in the skin, move the underlying fat and muscles, and then inject lung epithelial-derived tumor cells with mutation in KRAS and P53 in the left lung of immune-competent mice. On the right bottom here, you can see that a single tumor develops, which is similar to what we see in humans. However, in contrast to these subcutaneous models, the tumor is not visible by eye and accessible by caliper measurements. Therefore, we used a microcone beam CT imager to detect early tumor take in these mice and also detect the tumor development. To quantify tumor volume from a CT image, normally manual segmentation of the tumor volume is required. But you all can imagine that it is very labor intensive and time consuming to manually delineate all these tumor volumes for each mice. And therefore, we trained a deep learning algorithm on 60 manually delineated lung tumors to fully automate this task. Validation of this deep learning model shows that there is a high correlation between the automatic segmentations on the y-axis and the manual segmentation shown on the x-axis. And this correlation shows an R-square of 0.93. On the right bottom, we see the dissimilarity coefficient. And the dissimilarity coefficients indicate the percentage of overlap between the automatic and the manual segmentations. And then we can see that there is a high agreement between the manual segmentation, so the training data set, and the automatic segmentations derived from the algorithm. There are only with some really small tumors we see that there is some disagreement. So subsequently, this deep learning algorithm allows us to highly accurate monitor the tumor volume of these mice over time with minimal operator involvement. And again, we see that there is high overlap between the manual segmentations indicated in blue and the automatic segmentations indicated in red. In addition, this algorithm is also a very helpful tool to optimize the randomization of the mice of normal experimental design. Using a similar approach, we previously developed a deep learning algorithm for the automatic quantification of lower hind limb muscle mass in mice. Combining both of these deep learning algorithms enables us to parallel monitor the tumor volume and muscle mass over time. And here you can see the data from one cachectic mice, and we really nicely see that the tumor volume is increasing, and after around 3 weeks, we see that the muscle volume is decreasing over 2 weeks. And using these tools, we're able to fully optimize this model to study cancer cachexia. In the upcoming slides, I will show you some data on the characterization of the cachectic mouse model, but due to the lack of time, I will mainly focus on the biochemical data derived from the skeletal muscle. Approximately 70% of the tumor-bearing mice develop cachexia, and the cachectic mice have a median survival of 33.5 days and show significantly body weight loss, starting approximately around 21 days after surgery. Similar to the body weight loss, we see significant loss of muscle mass over time measured by CT. And if you look more into detail into the muscle wet weights at the end of the experiment, we see that this muscle mass loss is most prominent in the muscle plantaris, muscle gastrocnemius, and in the tibialis anterior. And the cachectic mice, in the cachectic mice, we also saw a loss of muscle function measured by grip strength. And important to note is that this grip strength is significantly correlated with the muscle mass in these mice. The observed muscle wasting in these mice is associated with the increased expression of atrogin, or the E3 ligases, atrogin and MERV1, but we didn't show an increase in NET4. The expression of NIP3 and GABARAP-L were upregulated, suggesting that there is an increased activation of the autophagy lysosomal pathway. And in addition, we also saw an upregulation of the mRNA expression of FOXO1. And together, these data indicate that there is an increased proteolysis signaling in the atrophied muscle of these orthotopic lung cancer mice. On the other side, we also see that the muscle wasting is associated with a decreased activation of AKT and P6, but also with 4-EBP1, which is not shown on this slide. And combined, this data indicates that there is a decreased protein synthesis signaling in the muscle of the cachectic mice compared to the sham control. We also found that there was an increased expression, or that there was an increased mRNA expression of GLUL and KLF15, and they are both markers for increased glucocorticoid signaling. We also found increased expression of myostatin and E-kappa-B alpha, suggesting that there is an increased inflammation in these mice. I started this presentation that orthotopic mouse models will be more relevant than the subcutaneous models, but of course we also were interested to see whether there are also overlapping processes visible in our newly developed orthotopic mouse model compared to the non-small cell lung cancer patients. So we performed RNA sequencing on muscle biopsies from the vastus lateralis of cachectic patients versus healthy controls, and also on the sham mice versus the tumor-bearing mice with cachexia. And subsequently, we performed pathway enrichment analyses on the downregulated genes. And then if we look at the upregulated or the significantly differentially regulated genes, or pathways, we see that mainly pathways related to metabolism, oxidative phosphorylation, and mitochondrial function are involved, and we see that the same pathways are highlighted in the mouse model versus the human patients, and we saw the same in 3 different types of databases. Next, we also looked in some microRNA expression, because for the same patient group, previously we did some microRNA profiling, and we look now into the most promising upregulated and downregulated microRNAs, which we saw in these cachectic non-small cell lung cancer patients. And in this mouse model, we see the same expression profile of these microRNAs compared to human. So to conclude, we developed an orthotopic lung cancer mouse model to study lung cancer cachexia. We developed novel tools for the automatic quantification and evaluation of tumor volume and muscle mass. The observed muscle wasting was accompanied by alterations in the regulation of protein turnover, reflecting increased protein degradation. And the orthotopic mouse model recapitulates key aspects specific to cachexia seen in lung cancer patients. And I would like to thank my supervisors from the respiratory medicine department and all their fruitful meetings on this work. And I would like to thank the Department of Radiotherapy for their help in the AI work, and the Department of Toxicogenomics, they mainly help for the RNA sequencing, and the Department of Precision Medicine for their help in the development of the mouse model. Thank you for listening, and I'm happy to answer any questions.
Thank you for the great presentation. If people have questions, go ahead and post those into the Q&A box. We do have one question. Are the tumors implanted in the middle lobe? How long is the recovery? And is there a large variation in the size of the tumors in each cohort?
Yes, thank you for the question. So we optimize the model now to inject the tumor cells in the left lung lobe. So in mice, on the right, they have 4 lung lobes, and on the left, they only have 1 lung lobe. And to reduce the variation also in tumor size, but also tumor take, we change from the right side to the left side, because we saw when we did the injections on the right side that the needle appeared often in between the lung lobes. So then we got a lot of tumor development intrapleurally, which was something which we don't like, and it also results in very rapid tumor growth over there. Related to the variation in tumor size, now we quite optimize the model and also the surgery technique. So now we see that the tumor development is quite heterogeneous now in all mouse, but it's less homogeneous compared to subcutaneous models, of course, because during the injection you have the movement of the lungs, and there are a lot of factors involved which can create some variation in this model.
Do you see a lot of metastases in this model?
Actually, we don't see a lot of metastases. So mainly we see the primary tumor in the left lung. Sometimes, but it's not really often, we see some metastases in the same lung lobe, and really some not often in the on the right side. And I think sometimes you see some metastases subcutaneous, but I think that's more related to the technique, so that we have some spill during the surgery.
Okay. And then we have one more question. In your work to develop this model, did you by any chance also develop orthotopic models that do not develop cachexia?
Yeah, so our main aim was indeed to develop one model which resulted in the development of cachexia, and one which was not resulting in the development of cachexia. So we have one other cell line, and if we inject that one, only 25% of the mice develop cachexia. So we see a clear difference, but still some mice are developing cachexia. So we did not find the model yet which did not develop cachexia, but I think it's similar if you see to patients in all type of cancers. There is a small percentage of people or patients which will develop cachexia. So maybe it's also something which is reflecting the biology in that way.
Great, thank you. Are there changes in food intake in your model?
We don't really see cachexia or anorexia in our model. If you saw the body weight loss, you see that if we really go till human endpoint, that the mice, sometimes you have cachexia for like 1 to 2 weeks. And we see that the food intake decreases like the last 1 or 2 days before human endpoint, but that's really because they get really ill, and that is most of the time due to the tumor, that there is some obstruction or something like that. But we don't see that there is anorexia during the beginning of the cachexia process.
Great. Thank you to all of our speakers. Everybody did a wonderful job. And just like our past Next Gen sessions, this year's Next Generation session participants will receive complimentary registration for the 7th conference. With that concludes our session. Thank you for joining us. We have a break, and we'll be reconvening back at 1:30 p.m. Eastern time